The protein that I chose to research was the Huntingtin protein. I chose this protein because it's one of the largest proteins in the human body, and its size and complexity present many challenges for researchers, especially with its mutant state. The mutant Huntingtin protein is the primary cause of Huntington's disease because it contains an expanded stretch of glutamine repeats, which I thought would be super interesting to learn about. As I stated earlier, the Huntingtin protein is a very large, common protein within humans. It plays an important role in nerve cells and neurons within the brain. It is essential for normal development before birth and found in many of the body's tissues with the highest levels of activity in the brain. Within cells, this protein may be involved in chemical signaling, transporting materials, binding to structures, and protecting the cell from self-destruction, also known as apoptosis. Some studies suggest that it plays a role in repairing damaged DNA as well. So this is a very important protein within our bodies. However, if it is mutated, it can cause Huntington's disease. This disease is inherited and caused by an expanded, unstable trinucleotide repeat within the Huntingtin gene, which translates as a polyglutamine repeat in the protein product. A polyglutamine is a group of neurodegenerative disorders caused by expanded cytosine, adenine, and guanine repeats. So in the normal protein, these repeats are usually 10 to 35, but if they are repeated more than 35 times, then the protein is considered a mutant. These images show kind of how the mutant protein works. So right here, it's normal, but right here you can see that there are multiple repeated CAG portions, which cause it to be extended. And these the extra repeats cause a neuron degeneration, which ends up causing Huntington's disease here. And as you look at the brain, they are significantly different. This says the frontal horns enlargement of the lateral ventricles are significantly larger. The primary structure of a protein is a linear sequence of amino acids that make up its chain linked together by peptide bonds. The Huntingtin protein is encoded by the HTT gene. This slide shows two sequences which represent the two chains that make up the Huntingtin protein. The primary structure of the Huntingtin protein includes a varying number of glutamine repeats near its end-terminus. This repetitive sequence is what makes the Huntingtin protein susceptible to expansion mutations leading to Huntington's, as discussed about on the previous slide. These sequences continue for thousands of amino acids, forming the complete primary structure of the Huntingtin protein. The secondary structure of a protein refers to the local folding pattern that emerges within segments of the protein chain. The two most common types of secondary structure are alpha helices and beta strands. These structures are held together by hydrogen bonds between the carboxy and amine groups that are not part of the peptide chain or peptide bond. In the case of the Huntingtin protein, which is a large and complex protein, its secondary structure is not fully characterized, especially considering its size and the fact that it contains regions with repeated glutamine sequences that can affect its folding. However, it has been found that it is almost entirely comprised of alpha helices, as seen in this photo by the blue. Among the folding patterns, there are also sections that don't follow the typical structure or bonding patterns. These areas are called random coils, as seen here in the pink, and they appear at the ends of the protein chain and in loops between the more organized segments. Random coils can vary in length and typically range from 4 to 20 building blocks. The tertiary structure of a protein refers to the three-dimensional arrangement of its entire polypeptide chain, including how various secondary structural elements fold and interact with each other to form a functional protein. The Huntingtin protein has numerous interactions between the R groups and the amino acids. The yellow dotted lines represent hydrogen bonds, which are formed between polar or charged amino acid side chains or between backbone atoms, contributing to the stability of the protein's folding structure. The disulfide bonds are represented by the circled spheres, but are relatively rare in cytoplasmic proteins like Huntingtin. These proteins are covalent bonds that form between the sulfur atoms of two cysteine residues, stabilizing specific regions of the protein structure. Huntingtin also contains hydrophobic interactions, which occur between nonpolar amino acid side chains, causing them to cluster together in the protein's interior, away from the surrounding aqueous environment. It also has ionic bonds, and these bonds form between oppositely charged amino acid side chains, contributing to the overall stability of the protein structure. The quaternary structure of a protein refers to the arrangement of multiple R group interactions between different polypeptide chains. Huntingtin is primarily known to exist as a monomeric protein, meaning it does not typically form complexes with other copies of itself to create a quaternary structure. It is a large protein encoded by the HTT gene, and it is primarily found as a single polypeptide chain within cells. While Huntingtin may interact with various forms and cellular components to perform its functions, it does not typically form high-order structures involving multiple copies of itself. And here are the articles and photos that I used during my research of this protein.